a wonderful problem to have that we will deal with and do a better job of next time. It is my pleasure to welcome my colleague, Dr. Eleanor Haglund, our first seminar speaker of the year. She's going to be telling us some very interesting stories about proteins that fold in knots. Hopefully there'll be a little bit of material for all of our various audiences to sink their teeth into. Eleanor and I met while I was a postdoc at Penn, and she came to Penn to do some NMR with another colleague of mine who's now a professor down at VCU. One of the great things for all of you that are in school right now or in grad school is that as you get older, all the people that you're seeing around you now become your friends that are elsewhere, and you get to go visit them. Eleanor was just telling us about how she's in the midst of now a trip that involves a total of 13 talks at different places, which is super duper cool. She comes to us from University of Hawaii, so she's come quite a ways. She's going to tell us a little bit about her academic path, but I think the main thing that I find exciting about Eleanor as a scientist and her work, to me, she does the kind of work that I do the right way. She doesn't. She's a protein biochemist, protein biophysicist like me, but she artfully mixes both theoretical and experimental work to draw a really comprehensive picture of how proteins behave. And I admire that greatly about both the work that she has done and the work that she's doing now. So please join me in welcoming Eleanor, where I'm excited to hear about her science.
Thank you. Wow, thank you so much for that nice introduction, and thank you for inviting me and making this day and tomorrow really awesome. The title of my talk is The Folding and Function of Proteins with Complex Topologies. Nucci told me that we have a mix of people from biology, physics, and biochemistry. Basically, we're going to talk about how do I tie a knot, and I think everyone can relate to that. And also, feel free to interrupt me at any point of this presentation and just ask a question. So let's see if it works. So I was also told, tell me something about your career path. You can hear I have a little bit of an accent. I did not grow up in this country. So I grew up in Sweden, that is over here. It is freezing cold and dark most of the time, so I live in Hawaii now for a reason. So I grew up in a town by the Blue Dot. I went to my undergraduate university north in Sweden to south of the Arctic Circle. It snows to cover house overnight. You see the northern lights. And I started my career in a research lab up there. The professor, Michael Oliver, that I worked for, moved his lab to Stockholm, which is more in the area where I'm from, over here. Then I got the opportunity to do a research project in Bangalore, India. So I went there to learn how to do FET experiments. My graduate work is based on how do proteins fold. Simple fundamental research questions studying a simple two-state model that we'll talk about later in this presentation. So we didn't care about biology at all. In my world, it's more interesting to do folding and function. So we're trying to understand how proteins fold, to understand how they function. So to do so, I went to UCSD working with Dr. Jose Anochik for the Center of Theoretical Biological Physics, learning how to do structure-based molecular dynamics simulations. After that, they moved the center to Rice University. So I spent 3 years there before I got my position in Hawaii. So the outline of my talk is introduction, first of all, what is protein folding? Why do we care, right? Complex topologies, what is defined as a complex topology? That is a really complex question that I can't answer today, but we refer to it today about knots and entanglements in proteins. I discovered something that we call Pierced Lasso Topologies when I was a postdoc, and that's why I have the job that I have today. I will talk about the model proteins we use to study these PLTs, and I will end it talking about threading of covalent loops and a short little conclusion before we can end this presentation. So I guess most of you heard about the central dogma, which is in biology, how does DNA get transcribed into mRNA that goes from the nucleus out to the cytosol? It gets caught by a ribosome, so we can do translation on the ribosome. We end up with a nascent chain that somehow has to fold most of the time into three-dimensional structure to function. After it's done its course, we have to degrade the protein so we can recycle the amino acids. So today we'll work on what happens here. This little step is what we'll talk about the most. The field of protein folding started a very long time ago when they discovered proteins and they learned how to crystallize things. But two really important events was, first of all, Leventhal's paradox. And he said, if protein folding is a random search through conformational space, it would basically take forever to fold. So here's this little thought example. If you have a protein of 100 amino acids, and every amino acid has to turn 2 different conformations for the protein to fold correctly, it would take longer time to fold one protein than it took to form the universe. So he stated that protein folding cannot be a random search. We have to have a code somewhere hidden in the amino acid sequence for the protein to know what the 3-dimensional structure should look like. A very important issue is then the amphissin experiment. Many people think once a protein folds, it stays folded throughout its lifetime, which is not true. It depends on the global stability. So we'll have a bunch of unfolding and refolding events throughout the lifetime of the protein. So amphissin did his experiment that probably a lot of you have heard about. He took ribonuclease A, he tested the biological activity, he unfolded the protein with chemical denaturing and reducing agents because this protein contained disulfide bonds. He then let the protein refold and tested the biological activity again to prove that a protein can actually unfold and refold reversibly in a beaker without the help of chaperone. So I always say this is kind of the beginning of our field. Me and one of my graduate students built this little historical plot. Proteins were discovered a very long time ago, and there's some events that we feel has more implications in the field than others. And just to mention a few, we have myoglobin was crystallized. The protein database helps a lot. And like I said, most proteins have to fold to function. So for us, it's extremely important to understand what the 3-dimensional structure looks like. With experimental techniques, we can use cryo-EM, we can use NMR, we can use crystallography. For various reasons, certain techniques works better than others. Also, one big change happened here a couple of years ago when they invented AlphaFold. AlphaFold is a prediction tool, and we want to emphasize it's a prediction tool that predicts what the 3-dimensional structures looks like. It's phenomenal when it comes to small, simple systems. One thing that AlphaFold is not able to do, 40% of proteins do not have a 3-dimensional structure. But with AlphaFold, every protein will obtain a 3-dimensional structure. But it's a great indication, and they do have a probability index, so we can see how likely is the structure to form. And it's extremely helpful in the type of research that we do. So I also want to point out, if we understand how proteins fold correctly on a molecular level, we can understand what happens when things go wrong. Most of you have probably heard about Alzheimer's disease. The tau protein aggregates, form fibers and misfolds, and it would kill the cells in the brain, and you develop Alzheimer's disease. ALS is another one, Creutzfeldt-Jakob disease, Huntington's disease. There's a bunch of proteinopathies that we know of today. If we can understand the correct folding pathway, we can also understand molecular steps on this pathway where things go wrong, and we can target those steps with therapeutics in the future. So how do we do this? In the simplest case, if we look at a free energy profile, like any chemical reaction, we need to climb a free energy to reach the lowest energy state in nature, which is the native state. The unfolded or the denatured state has lots of entropy, lots of different conformation states that we can have. Once you find the right context to gain the energy to climb up the transition state, a protein would fold. It's driven by the hydrophobic effect, where the protein tries to hide the hydrophobic residues in the center of the core. And once we hit this state, it's downhill folding to the native state. In the 90s, Dr. Jose Onochik and Dr. Peter Wallace came up with a folding funnel model, where the top of the funnel represents the random states of an unfolded protein. We start to find the correct context to kind of drop down into the funnel, and depending on the ruggedness, we will reach the final native state. This is the simplest case where we have no intermediate states populated on the folding free energy landscape. So we go from a simple unfolded to a folded state. We call that a 2-state protein, and we can study those. But life is never that simple, right? That's in the simplest case. So what can increase the ruggedness on the landscape? One thing that was discovered in the 90s was that protein can actually self-tangle. It took about 10 years for people to accept this. So when we look at it, I'm going to pull up the next slide too. Mathematical knots have been studied for roughly 150, 200 years. So this knot diagram is 150 years old, I believe, today. And in mathematical knot theory, we have to have a closed circle. So this is the simplest knot. I don't think you and I would call this a knot, but it's called an unknot. And depending on the entanglements, we reach the simplest second entanglement. It's a 3-1 knot, where you actually cross the backbone 3 times. The nomenclature is mentioned how many times do the chain cross itself. And what does that mean? In proteins, we don't have a closed NNC terminal. So what we do is we look at a protein and we pretend that they meet somewhere in space. And if I do something like this, and I pull and I tug and whatever I do, I cannot change the topology of this knot. It's always going to be a 3-1 knot. That's the proper definition of a knot in a protein. Because no matter what I do, the knot cannot fall off the chain, right? So talking about that, in the 80s, Connolly and his cohort, they came up with a model where they say sometimes a protein can find a contact here, where the protein actually self-tangles and backs through this covalent loop. And this was the beginning of what we call Pierced Lasso Topologies today. It is a little bit different than the regular knotted proteins. Like I said, the first knotted protein was discovered in 1994, and it was a 3-1 knot. It took about 10 years for people to accept this, because they say, how can a protein do that? So for real, true knotted proteins, what happens is you have the polypeptide chain coming off the ribosome, it twists itself one time, and then you pull something through it, and then we connect the NNC terminus, and that's how we have a knotted protein. More complex knots can actually be developed if I just twist a few more times, right? And now I have more than 3-1 pass. So those are true knotted proteins. There's something called a slip knot. That's basically what you do with your shoelace, because you thread something through and you create a knot, right? But if I pull, it's no longer there, even though the NNC terminus is connected. So that is not a true knotted protein, it's a slip knotted protein. There's another class of protein that is called lacopeptides. They're small, 16 to 21 amino acids long, and they have an isopeptide bond here shown in red, where part of the structure is threaded through. These little lacopeptides are expressed or function in bacterial proteins, and they are co-expressed with an enzyme that can make this bond. Without it, we cannot do it. And that's a little bit different from PLTs that I will show you in a few slides. The other version contains cysteines just like ours. It's called a cystine knot. They're super stable proteins because they have 3 disulfides. So if you can see here, same thing here, the blue is kind of the covalent loop, and the red is the thing that threads through. But for those of you who knows how to read these structures, this is actually not a backbone. It's just the side chain of 2 other cysteines, right? So being an experimentalist, this protein, and to study threading of proteins, these proteins are great, but how do we get the linear protein? That is a complication, because we have to mutate it and change a lot for the protein to get something that is not tied into a knot. How do we prove that the knot is there? Because again, with experimental techniques, if I do thread, there's not a huge difference by adding a knot. So that is a big conundrum in the field of knot theory in proteins. How do we know that the proteins actually untie and reach a fully unfolded untied state? With this case, I cannot just change the chemical environment. I have to break that or not do it. So we're hypothesizing that these proteins actually fold the red part in, put it in place, and then you clip it in. We kind of call that a belting mechanism, which will happen the same thing with cysteine proteins. A lot of proteins with cysteines are secreted proteins. So they go through the air Golgi, where you have an oxidizing environment, you have thioredoxin, glutathione that can promote the bond and make and break the disulfide. So the belting mechanism, again, is just when the thing comes in place and I add the cysteine around it, just like a seatbelt. So we hypothesize all our proteins actually fold like that. The question is what happens once I'm secreted. Because once I'm secreted, most likely I will unfold and refold just like regular proteins. And the question is, can I thread when I'm under an oxidizing environment and I have that loop? For this protein, we also cannot control that because we cannot specifically treat one. So you will either have that circle formed by 2 disulfide and the threaded element being a disulfide bond, or you don't have it. So one thing that I learned when I was a graduate student, I got a lot of questions from the chair of the department about disulfides, and my protein didn't have any, so I didn't know a lot. Which is kind of funny because that's all we do today. So I thought a disulfide bond was just something that changes the free energy locally where you have it, because we do add a peptide bond. So if we add a peptide bond, we restrict that part of the structure when the bond is there, and you get more freedom when the bond is not there. But I never thought of it like circles. So if you have 2 cysteine at the N and C terminus of the protein, they will actually form the same thing I told you, mathematical knot. It's called a 0 knot or an unknot. The other possibility is that my disulfide bond or my cysteines are somewhere in sequence. I will form a cinch-like topology, and the size of the loop, of course, varies depending on the sequence separations between the 2 cysteines making the disulfide bond. And the last variant is a lasso, when one of the cysteines are either at the N or the C terminus. So this actually happens in every single protein that has a disulfide bond. There's nothing unique about that. What happens is when something actually pierces through the lasso, that's where the fun starts. And this is what happens in the proteins that we work with. So I studied a protein called leptin that we'll talk about, and that's the first time I saw this topology. Just to bring up disulfides, first of all, like I said, they are strong covalent bonds. They happen outside the central dogma. They're post-translation modifications. They may form threaded topologies, and they can promote or obstruct the formation in different cell compartments, because the cytosol has a reducing environment while the extracellular matrix is oxidizing environment. And this can tweak the conditions depending on the environment, but also the existence of pyridoxine and glutathione that can be driven by molecular oxygens. Thinking about human health, if we have cancer or inflammation, we do change that in hypoxia and hydroxia. So we think maybe this has an important role in biology. So post-translation modification of disulfides occur beyond the central dogma. But the question is, if I found this in one protein, why should you care at all? Right? If it's a rare coincidence. So together with my former coworker, Dr. Joanna Solkowska, who is now at the University of Warsaw, she came up with an algorithm where you can actually search through the PDB, and I'll show you the algorithm in a little bit. So we searched through the PDB and we found more than 600 proteins which has these PLTs. They're classified into 14 different classifications, and they do exist in all kingdoms of life. I also want to point out the plots that you see are potentially biased by the PDB, because we know not all proteins are crystallized. Maybe human proteins are more exciting than a plant protein, what do I know, right? And we see that they have 14 different classifications, where hydrolases is one of the bigger classes, but also immune system proteins. And we all know this COVID spike protein is actually a PLT. And we see that they do exist in all cell compartments with a dominance in the extracellular matrix where we have an oxidizing environment. So we hypothesized that PLTs may act as molecular switches important in human health, because the chemical environment will determine if I have a threaded topology or not. They are found in many human proteins. They exist in cell signaling proteins, bacterial proteins, and viral proteins. And I just did a quick little word logo plot and saw that they exist in many proteins that we know are part of proteinopathies influencing human health. So the big questions we ask ourselves when it comes to entangled protein structures. Many people say that threaded or knotted topologies exist because of stabilizing effect. It can have controlled biological activity, and it can, how can we determine how they thread? So these are the fundamental questions. We're going to see if we can answer some of them today. To do so, we study 3 model systems. I'll do this. So first of all, we study leptin, like I told you about. Leptin is the satiety hormone that tells you stop eating, you're full. So in terms of human health, it plays an important role, not only in obesity, also in cancer, diabetes, and many other sequential disorders to obesity. It has an L1 topology where the backbone threads through that loop once. We have superoxide dismutase, and I'm showing the protein for M tuberculosis, so it plays a role in tuberculosis. It has a larger loop, and you have 1 crossings again. The gray surface is this algorithm that I'll tell you about. So it's just a triangulation to show you a plane where it's easier to see what is on either side of the protein. We also studied the human protein that exists in the cytosol, because we want to ask ourselves, how come you have a protein that has the same functionality to convert the reactive oxygen species to non-toxic species? One is in the cytosol, we have a reducing environment, and one is in the periplasm of bacteria. So when they invade your lungs, your body will spit ROS at them, and they just convert them to non-toxic species. So we're interested in the sequence evolution in terms of that as well, and what is the role, because this protein contains the copper for the chemistry, but also zinc if you exist in the cytosol. This protein that exists in the periplasm don't have the zinc ion. So we think that's an interesting project to understand sequence evolution of 2 proteins that evolved in different environments in terms of looking at the Pierced Lasso Topology and the disulfide bond and the role of metals with that. The last system are chemokines. Chemokines are small 70, 80 amino acid proteins. They have 1 disulfide bond down here. They have 2 crosses of the threaded elements, it's an L2. And they also usually work, they can activate the GPCRs either as monomers, homo, or heterodimers. So we study, I think, 7 of the ligands in 2 different receptor systems, because it's really fascinating systems that I'm not going to talk about today. but the biology, they can activate more than one receptor, and it's a very promiscuous system. So what do we do with this? The interesting thing with PLTs is we can just mutate or substitute amino acids and move that disulfide bond around. So if we look at leptin and MTSP, we have this kind of scenario. We have a disulfide bond, one threaded element. If I flip this one to the terminal end of the protein, I can form the zero knot. If I add a disulfide, this protein actually has a second disulfide holding it in place. So when you unfold that protein, it will never unthread, while the other two will, hopefully. So we added this disulfide link in leptin to see if we force it to be threaded, does that change things? And we can reduce the protein for most of them, but not all of them. So that's the little model system that we have, and that's the difference in topology that I'm going to discuss today. How do we do it? So in my research group, like Nucci said, we use structure-based simulations. So to do that, we look at this Hamiltonian over here. We can look at coarse grain models, we can do all atom, and we can do C-alpha atoms. So what I want to talk about today is basically C-alpha simulations. So we look at the two bonds between two atoms, we look at the angles and the dihedrals between them, and each amino acid is represented with a single B. We also do NMR, kinetics and thermodynamics, studying circular dichroism and fluorescence. We're about to build our own cell lab so we can do the activity assays and see what's the biological role of switching the loops around. We do some cryo-EM and TEM. And recently, we just created a little fruit fly in Hawaii that we actually knocked out the leptin amyloid protein. So we can study these loop variants in fruit flies and look at the phenotype. And we actually made the fruit flies obese. They gained about 1 gram. So we can also study obesity in these fruit flies. And for those of you who works with microbiome, we also study the microbiome, what happens if we knock leptin out. Interestingly, they get a more diverse microbiome, which is not what we expected. So this is, again, what leptin does. It's made in adipose tissue. It has to get out from the cells. It's secreted in secretory vesicles. It travels to the hypothalamus where it activates a receptor to do an internal signaling, and it controls food intake and energy expenditure. So the first question we raised, coming back to the fundamental question, leaving biology for a little bit, is for this protein to work, we took the oxidized protein. Oxidized means the disulfide is there, and we have a threaded element. And we ran the thermodynamics using fluorescence in this case, and we reduced the conditions and broke the disulfide. This is an equilibrium titration, so we use a chemical denaturing, and we look at the fraction of unfolded protein normalized for these proteins, because they have different signals. And we can look at the regular threaded protein. It has a stability of around 6 kilodaltons, 6 kilojoules, I think. And then when we reduce the disulfide, unfortunately, this just keeps going. We did follow-up experiments, and so far we still have not, unfortunately, found a condition where this protein is folded. So for leptin, we can unfortunately not study the reduced protein. It's never folded at 25, 37, or whatever condition we tried. We tried a lot with reducing agents, trying to get an NMR spectrum. It just reoxidizes all the time, or it aggregates and falls out of solution. So we can't study that protein. In terms of stability, we can form this type of knot and say, does the threaded part of the protein influence the stability? And to answer that question, they overlap pretty well, right? So we can say that in terms of PLTs, by just flipping the disulfide, it seems like the cysteine is the one maintaining the stability. It has nothing to do with the threaded element. And there's studies saying both for knotted proteins. Some people say that knot is not there for stability. Other people claim it's there for stability. But in our case, we can easily do that experiment and say, hey, they do fold, or they do have the same thermodynamic stability. So it is not something that affects stability. The other thing we did, because we cannot get the reduced protein with experiments, we turned to simulation. This is actually an all-atom simulation, but we represent it on a coarse-grained C-alpha. So every bar in here is the amino acids in leptin if you look at this spot. You see the cysteines are here at the C terminus and residue 96. And we plot the reduced protein versus the oxidized protein here. The yellow here just shows the difference between the two. So at the top, we have the sequence, we have the secondary structure, and we have an amplitude of the fluctuations. So we can see how much does it move. And interestingly, the oxidized protein is more dynamic than the reduced protein, but the overall trend is we don't just locally change things around the disulfide. You can see that the whole protein moves differently in solution. Yes,
This is from your C alpha.
yes, it's an all atom
is-
simulation, but coarse, yeah,
Okay, so what we're looking at here is the amplitude of mobility of the backbone only.
yes, in this plot, yes.
Okay.
So we see this shift between the two. So we said, what about if we study other helical bundles? Because that's what we wanted to start. They have similar secondary structure, or they have identical secondary structure, similar folds, similar motifs. So we took a few proteins that have PLTs, a few proteins that do not have a PLT, and we did the same simulation. So this protein has a cinch. The cysteine is formed between, the disulfide is formed between two cysteines somewhere in sequence. And you have an open end and C-terminal where nothing threads through the covalent loop. And when we do this, we see that the reduced protein and the oxidized protein is very similar, except where we have the disulfide. We do gain some conformational dynamics again because we don't have the peptide bond holding it in place. And this is true for all the 12 proteins that we tested. So we said maybe the conformational dynamics has an effect on biology because how I move if I have to interact with something has an important role. So we hypothesized that the conformational dynamics, this is again NMR over here for the oxidized protein, but this is a structure-based model, is not an NMR structure that I'm showing. So you can see that this protein is really malleable. One of the reasons we believe it's malleable is that you have 6 isoforms of the receptor, the full-length one in your brain up to a soluble one, 7 domains, just floats around in your extracellular matrix, so that you can interact with this protein. Not that we know what all receptors do. And it seems like if we have this threaded topology, and we have something threaded through, it has kind of a tsunami effect. I don't know what to call it. If you have rotations in the disulfide and movement in this loop, half of the protein is on either side. So if this loop is moving, it makes the whole thing move more compared to if you have an open structure, it moves less. But we can unfortunately not test that experimentally. So the next question is, how do we thread? So again, if you have this closed loop, the interesting part with leptin, to do this experiment, it turns out it's the perfect model. So our loop is 50 amino acids in the native 3-dimensional structure. I have 50 amino acids on this side and 50 on this side. So the question is, how do I reach my threaded native state? So there are 2 pathways, right? I can either take my terminal end and do something called plugging, because now we can't do the belting mechanism anymore because we have the disulfide intact at all times. So I can plug it and reach my native state, or I can slip knot it and reach the same native state. So in terms of probability, it's the same. No matter which pathway I take, I have to drag 50 amino acids across. Again, this is extremely hard to obtain experimentally because we don't really, even though we can use fret, am I on this side or on this side? It's really hard to get the dimensions. So that's again why we started doing computational work first to see what we can learn and then see what we can do in the lab. So again, this is the protein. Q is the number of nativeness from 1 to 0. 0 means no native contacts, 1 means all native contacts. We have the time, which is an arbitrary time because it's a structure-based model. And then we have here the amino acid sequence and the time it takes to cross. So there are 2 pathways, like I said. If I start with the N terminus, it will be a low number going through first. If I slip knot, it will be the number around the second or the disulfide bond up here. So if I go the plugging route or the slip knotting route, it turns out they reach the threaded point at the same point. So we can hypothesize maybe the 2 events take the same number of time because we have to drag 50 residues across. Doing some math here, we figured out that 99% of the time this protein actually slip knots through, and 1% of the time it plugs its terminal end through the loop. So in the lab, the big question again is, do we know if this protein actually unthreads in nature? Doing the computational simulation, we see that it fluctuates a lot, completely unfolded, but it doesn't unthread, collapses back to the native state, tries to unfold. After a few attempts, it will unfold and unthread. So to do this in the lab, there's no technique as far as we know where we can determine that. So we thought we could do something to see what we can do. So first of all, if you look at the top graph, we have this protein. Like I said, this is the wild type protein up here. 1 disulfide bond, 1 threaded element, and it's the red graph you see here. Kinetics, for those of you who are not familiar with a chevron plot, we take chemical denaturing and we do about 35 experiments. Each dot is an individual experiment. So we take the native protein, we jump it into 8 molar of urea, and we monitor how long time does it take for this protein to fully unfold. and we can plot the kinetics for that point. We use a logarithmic scale because it gives you this nice V shape and a linear relationship with denaturants. So we can actually extract the unfolding and refolding kinetic times for that. And we do extrapolate to water. We don't want any effects of the chemical denaturants used or anything else. So we extrapolate to water. So in this little window, we also show the circular dichroism spectrum. For those who don't know, it's a circular polarized light. Depending if on beta sheet or alpha helical, we will have a typical spectrum of the two. So an alpha helical protein will have two minima, one at 222 and one at 208, 209. And that's what we see with both wild type protein and the double link protein to say that we didn't perturb the structure by adding a second disulfide. So for the blue protein, we put this disulfide in here to make sure that it never unthreads. So basically it's just a 4-helix bundle that can unfold and refold. So by doing this experiment, we see that if I have to thread, I slow down the refolding kinetics with about 100-fold. So we don't know, but we can hypothesize that maybe in this red plot, when we have a wild type protein, we do have a threading event that we can observe when we run this experimentally. But again, we can't say that we know that we have that. We can just hypothesize that because I slow down the kinetics by 100-fold, if I force it to thread, we can hypothesize that. The other variant, we just move the disulfide. So now we have it at the N terminus instead of the C terminus. And we keep the second cysteine. So the loop is now 100 amino acids. We do the same computational work. And we see that if you extend the loop, you prefer or you dominantly plug your termini through. Yes, we do plug a shorter piece than we do for this protein because we only have 146 amino acids to play with. So another project we're going to do is to shorten and extend the loop and see where the threshold is when we switch. Using other 4-helix bundles, somewhere around 65 amino acids is where you flip from a slip knotting to a plugging mechanism. The bigger the loop, the more likely you are to plug. So we did simulations on this, and we see that this one 95% of the time plugs through. So doing the experiment, same thing again, the circular dichroism spectrum overlaps pretty well. And doing the experiment, if we ignore the letter curvature, which can happen for various reasons, and we look at the linear parts of this graph, we can say that the refolding rate is basically the same, which would agree with this. There's no time difference experimentally by threading, or slip knotting or plugging. So we can't use that as a tool to say, do we do this or the other? The other thing is we do ensemble experiments. So how would we obtain data for something that happens 1% of the time? So we hypothesized that leptin threads and unthreads in vitro, which was a big issue in terms of what happens when I'm in the extracellular matrix. Does the protein actually unthread? And that was a really complicated issue for knotted proteins. And I don't think they've ever showed that they can untie a knotted protein except when they use mechanical force and optical tweezers to pull the protein apart. And that was done by Sophie Jackson and Matthias Reeve. Coming back to kinetics. So looking at 3 proteins, the gray one is just our dynamic disulfide. It's just a pure vanilla structure-based model. We don't change anything at all. So that means the disulfide bond can actually be made and broken. Experimentally, it's hard to measure things at the redox potential. So we split the simulations the same we do with experiments. So we have a reducing and an oxidizing protein. So if I fold my protein, and this is time steps, and this is counts how many times it happens, and we fit it to this Gaussian distribution, and we can see that the blue protein, which is reduced linear protein, it folds a lot faster and a lot cleaner. You can see the distribution of the tail, it just tapers off, and you don't have anything basically. Compared to the oxidized protein in red, we slow down the folding, more ruggedness on the landscape. We have a long tail, so you add ruggedness to the landscape if you add a threading event. When we analyze our data with computational work, we use, again, a Q, the nativeness of the protein, and we study the alpha atoms, and we measure F of Q, the free energy, and plot these data. So as you can see here, we see the denatured basing, low Q, the native basing, high Q, native contacts are formed, no contacts are formed, right? But when we do something called a p-fold analysis, where you should have 50% of the molecules should fold and 50 can unfold, that should agree with where our transition state is up here. It actually dips down into this basin. So I turned to my also former co-worker, Dr. Jeff Noel, who's now working for a company in Germany, and we did some simulations to try to figure out what is going on here, because Q is a great reaction coordinate for the reduced protein, but it's not a great reaction coordinate for the oxidized protein. So we used Joanna's algorithm. So what did she do? She takes a protein, and she used a triangulation of the minimal surface. So you can see the artificial triangles in there. So the first thing in her algorithm is to detect the covalent loop. The covalent link, it could be other things, like I told you, the isopeptide bonds. But we're interested in the disulfide link PLTs, which represents 90 or 95% of all known PLTs. So first you find a disulfide link, and then you actually span the surface, and then you detect where the crossing happened. So you see a blue and a green. It's just describing the directionality across the surface. And then you minimize the energy when you do this. And with this algorithm, we add a crossing coordinate, because topology matters for these proteins. So by adding a topological parameter to our analysis, we can see this similar plot that I showed before. We have QC alpha, we have Q, but we also add a third component to this. So we have 3 dimensions of a crossing coordinate. You can see that the population of state, low populated, high populated. So the mechanism, here's also the sequence. You can see on the y-axis, you can see one little pointer. So this is the plug-in pathway. And you can see that it's barely not populated because, again, we only have 1% to work. And the slip knotting mechanism, slip knotting pathway comes from the top and down. So by using 3 coordinates, we can split the 2 pathways. Doing a p-fold simulation, we can see that it also agrees here. So if we plot the crossing coordinate and we do our p-fold simulation, we can see that the protein actually has a transition state around 0.25 and not 0.45 that I have in the other graph. And that agrees really well with what we see over here, that we actually have a transition state barrier at a much lower Q. So why is that? So the threading part of the protein to thread seems to be the rate limiting step for this protein, and we barely have any native contacts. We can't really observe this experimentally, but what we can do, first of all, we can do a good prediction if we use a topological parameter of C combined with Q, and then we can actually map out the transition state in agreement with p-fold. So we can do something called a 5-value analysis to look at what does the transition state look like. Transition state is so high energy, so we can't really observe it experimentally, but we can do some manipulation here. So what we do is conservative mutation. This was a methodology that was developed by Dr. Allen first. So if we have a protein, we can do a substitution. We have to be careful. We look at hydrophobic amino acids that points towards the core and are present in a secondary structure, because loops don't really have a big role in folding. So we take those residues, we take all the hydrophobic residues, and we mutate them to something smaller. We want to maintain the entropy, so we don't touch prolines, we don't touch glycines, so we make it usually an alanine. To see what happens if I perturb the structure, I kind of make a hole, but keep the backbone intact by adding the short amino acid. So if this amino acid has no part in the transition state, can you see this? It's working, but I don't know why we can't see it. So otherwise I can find, there we go. So if this amino acid, look at the red amino acid here. If this amino acid is not part of the transition state structure, when we look at the wild type protein, when we mutate it, it will not play a part in the refolding rate for the protein. So we'll maintain constant. Showing you the Chevron plot again, that means that the refolding limbs overlaps between the substitution and the wild type. And that means that this red amino acid forms on the downhill pathway of the free energy. If instead I have an amino acid, the blue one, and I make a mutation of something that was important for this transition state, if I make it a whole, it will take a longer time for that protein to find its native context, and that will slow down the refolding rate, and we can plot that. So basically a 5-value analysis, we use the logarithm of the folding rate for the wild type minus the mutation divided by the whole perturbation of the refolding event between an unfolding event for the wild type protein and the substituted protein. So keep in mind, we study what happens when we mutate things, but we bring it back to what the original amino acid was important. Does that make sense? Awesome. So when we do this, we can do that computationally as well. We don't do mutations with computations because we can compare the energy of the unfolded state with the energy of the transition state instead, and we can convert that. So we did that for our reduced linear protein. We have a scale down here from 0 to 1 and some negative values in blue. So if you look at the reduced protein where we don't have a threaded element, you see kind of yellow spread out everywhere except for helix A, which is a diffuse nucleus, meaning that most of the protein is part of this nucleus that makes us climb the free energy barrier. That would give us the energy. So when the yellow ones find the context, then things starts to move and we can cross the free energy. For the threaded protein, we see that we barely have anything at all because most of it is white. We have some blue contacts, but keep in mind they're like -0.1, -0.3, or 0.03, 0.01, sorry. So they're barely negative, but we color them blue just to show. So basically you have two contacts in here. We didn't really know if that was true, so we did the same experimentally. So we mutated 20 amino acids, and we can see 2 yellow 5 values in here from experiments. So what does that tell us? We can conclude that most likely the protein has same thing as no context that would agree with our new crossing coordinate being added to the free energy profile, that we actually thread already in the unfolded basin. And that is the rate limiting step for folding. For true knotty proteins, they first have to form the stability of forming this loop and twisting. So then they thread. For Pierced Lasso topologies, they seem to thread in the unfolded basin, and they have very few native contacts in there. So by that, I would like to conclude and say that Pierced Lasso topologies do exist in all kingdoms of life. They have different biological functions, and they do exist both in reducing and oxidizing environment inside and outside the cell. We can hypothesize that maybe they act as molecular switches, turning the biological activity on and off. Most of them are active in the oxidized state, but there are examples where they're reversed, where the reduced protein is active. We know that from what we've seen theoretically with computer simulations, we can say that yes, the threaded topology introduces conformational dynamics in the entire molecule, not only locally where they're at, and we hypothesize that's what controls the biological activity for them to act as molecular switches. If they have a role in human health, possibly because they are in proteins that affect human health in both human proteins, bacterial and viral proteins. And lastly, we want to say that the 3D landscape, we need to start thinking about topological constraints in topology. Maybe it has a bigger influence. We just don't know any experimental technique that we can use yet to study this. You can do FRET, single molecule. The problem though is just with the orientation. Where do we put the FRET pairs? PRE is another possibility that we looked into. So that's where we want to go next and see how can we observe this experimentally. We have our 3 model systems, even though I only talked about leptin today. Maybe one of the other one is better, so we can use both the reduced and oxidized protein. Leptin folds within millisecond, which is not always the best when you want to do, if you want to do NMR or kinetics. By that, I want to conclude with a few more slides before I will end my talk here. So we would like to say that in Hawaii, we can contribute with research that has an implication on the islands and the state of Hawaii. We study tuberculosis and ALS, obesity, inflammation, and cancer. We do chemical education, and we do tip into knot theory a little bit. I was not the best undergrad. When I was an undergrad, I would come from the gym, show up, and they threw a lab manual at me, and they said, "You read it, right?" And I said, "No, yeah," and I didn't. And I feel like there's so many things we can do with chemical education. So I was lucky enough to get the NSF Career Award. So in this, I said, can we develop a web app or a tool instead of giving them a lab manual for the type of research that we do? So we have created a lab course. Instead of giving them a manual, we're going to give them a nucleotide sequence and say, figure it out. They can't just figure it out because they have never done it before. So we're going to have Q&As. We're going to have little games in there. They have assignments to learn how to use PyMol, for example, because most of you that have not worked with proteins may not think of proteins the same way I do. My background was actually in molecular biology, where a protein is a circle or a Pac-Man or a triangle. It didn't really have a structure. So the goal is to teach students the scientific mindset and create this way of thinking, trial and error. And it's only going to be try it, and they're going to test it in the lab. So it's called the Aloha Biochemistry app. We use the little ribosome and our little islands to connect it to ourselves a little bit. It's the ABC app. And then ACS contacted me and say, Eleanor, do you want to write a book about protein folding? So I said, okay. So we wrote a book. It's being peer reviewed right now. So we're going to have this book also included in the web app. So we do that. I like to engage the undergraduate student in my lab. So we have, by the way, this is our website, our group, my email. If you have any questions that you don't want to ask in here, please email me. So my undergraduates are responsible for Fun Fact Fridays. So every Friday, we try to post something, a little fun fact. We do it on Twitter and Instagram to get them involved in this mindset that you can't just write whatever you want. It has to fit in the format of Twitter or whatnot and talk about knot theory, talk about Hawaii, or other little fun facts that we can learn. And I can't come up with a good one, but they're really awesome in doing so, and I'm learning a lot from them. So by that, I want to thank the NIH, the NSF, and the Hawaii Community Foundation. This is my research group in Hawaii. All the Leptin work is basically done by Jennifer Simeon, who's a 6-year graduate student in my group. Grace is also working on Leptin. We have the chemokine crew with David up here, and Ivy is the SOD person that works with that. We have some collaborators, and I really want to thank you all for coming, and I hope you have some questions. And if you know someone who wants to live in paradise to go to graduate school, we have a graduate program, and you're more than welcome to apply and ask me questions if you want. And we really, really want awesome graduate students to come to our school. By that, I just wanted to say thank you.
In your simulations, do you see any rotamer preferences in the areas around the knots or where the disulfide linkages happen? And second part of that, are there any deviations from traditional structural elements where you might get little tighter folding of helices or unfolding of helices to accommodate the crossing?
Let's see if I understand you correctly. You mean the rotations in there?
Well,
the
rotations,
dihedrals?
the first part is rotamers of the side chain.
That's a great question. Most of our simulations are C alpha, so we don't really see the side chains. In terms of the rotations, I think you're absolutely right. So there's also part of the Hamiltonian. We do have the dihedrals. And one thing, I cannot get any of my students to do any simulations, and we tried with collaborators. And I think it's really important because, like you said, we get kind of, if you just rotate the disulfide, you kind of pre-slip knot it just by flipping the disulfide. And that's something we want to look into because we can never get it to fold with an all-atom simulation. We can only study unfolding. So I think it could be something with the simulation because experimental, we see it. Another thing with experiments people have talked about, we've thought about putting non-natural amino acids in there, but talking to an organic chemist, he says, what are you going to change? Because the whole flip is going to rotate anyways. Because I thought the stiffness of the bond could help in doing that. So I don't know, but it's a great question. And I would want to know if someone has an idea how to do it. I think it's really interesting because that also affects slip knotting versus plugging and how we move the entire conformational dynamics because you have a little twist down here and the whole thing is going to move more, right? Awesome question.
We have a request on Zoom for you to back up and leave the ABC app slide up. If there's people who want to go to see it. Thanks.
Awesome.
I'll ask a question too. So much of what you talked about, like in terms of both the free energy effects and the kinetics, scream to me entropy and the ability of the protein to explore its sort of thermal landscape, right? With respect to slip knotting versus plugging, how does the length of the tail influence that? The example that you gave us, there was sort of equal 50 amino acids on the loop side and 50 amino acids on the sort of sticky heavy side.
Yeah. For
lack of a better term.
But
so what happens if you change the size of that loop?
Yes. So we change the loop to bigger and then you plug. But I think the question is even better that you're asking, and that's something we want to do. Keep the loop 50 because we already determined, and I think most people, if you think about it a little bit, it makes sense that a bigger loop would plug than slip knot. So we want to both extend the end terminus of leptin and also cut it.
I guess what I'm thinking about is like
because that will probably change things.
If you start this, it seems to me like the slip knotting process would be more likely to start. Because the end of the loop that would have to plug is sort of flopping around.
Yes.
Right. Whereas the available conformational space, conformational entropy of the pieces that are closer to the tethered side are lower, right? So they're going to have a lot higher likelihoods. I'm picturing if their tendency is for them to do the same thing, once something plugs, it goes all the way. It's less likely to come out. Yeah. Is that right?
Yes, we do see also that it actually does that, whichever, because it's the rotation of that disulfide too. If you plug, you actually put one terminal across. If you slip knot, if I use my rope again, the slip knotting mechanism will start with two threaded parts, right? And then it kind of have to like flip that disulfide, so you have one, because the final product is only threaded once, not the two. So if that answers the question about that. Another thing I want to point out, we do all these 5L analysis and we don't see any destabilization by putting a shorter amino acid in. It is not what we expect at all. So we have to also study the thermodynamics. So it's a kinetic effect.
Why would you expect a shorter amino acid? So this was the second part of my question. You're showing us this example and doing a lot of your analysis based on the background, which with invisible side chains, but the side chains are there filling space.
So for experiments, we do the side chain, right?
Yeah, but I mean, like,
Well,
if you're talking about you have this loop that forms,
Mhm.
right? If you have a loop that's small enough, the side chains are sort of closing that space.
Yes,
Right?
for the other
But once
proteins.
you get big enough, there's sort of, I guess, water in there. And everybody thinks water just gets out of the way. Of course, that's not necessarily the case, right?
Right.
So to what extent does the conformational, I think it's some of what Greg was getting at. To what extent do you pay attention to, or you started thinking about the conformational flexibility of the side chains themselves in dictating the energetics and the kinetics?
That's also a great question. So looking at leptin with van der Waals radius and just the pi mole structure, they point towards the solvent actually. So it has a big pore, while the other proteins don't. And that's why we want to have a discussion with all the knot theory people. What is a complex topology? Because I think most of these
Wait,
proteins.
so what I'm sorry to interrupt.
Yeah.
What you're saying is that if you take one of these loop proteins and you pull the knotted section out, the piece of the protein that goes through the loop is the hydrophobic core.
So kind of, if you just take the pi mole structure and you look at the van der Waals radius, the side chains point towards the solvent and not towards the center. So in the case of leptin, it looks like it evolved to leave it open, but absolutely you have water in there. What else would be in there, right? And then when you thread something, you can kind of visually see it's gonna work. But in my book, when I look at chemokines that has the second disulfide, it's clogged in there. It's never gonna pull out. So that's why I'm always thinking about it is what is complicated, maybe because they're structures that have up to 6 crossing and they can have both the N and the C terminal, and they have like super coiling where they do all kinds of stuff. But the question is, do they really do that? And I think they're actually belting and they might not unthread ever because they have even a second disulfide, many of them, to hold that in place. So that's why I think it's interesting to study the L1 topologies. Superoxide dismutase has a 100 amino acid loop and only 9 amino acids being threaded through, and they plug dominantly, right? So in those cases, I'm pretty certain that the side chains do not point towards the solvent and they're pointing inwards. And it's 9 amino acids that go through in this threaded element. And depending on which bacterial protein or whatnot, that region is conserved. So it looks very similar if the disulfide is there. Does that answer your question?
Yeah, totally. And I have
Right?
a million more questions we can geek out later.
When you're looking for PLTs across domains and kingdoms, how confident are you when you find one? Is it based on primary amino acid sequence? And isn't that predicted from the DNA in the blast search?
Great question.
So we're a couple steps removed from
Right.
really finding PLT.
So we're looking at the published structures within the protein database.
Okay.
And then we use Joanna's algorithm. So we say, do I have a covalent linker somewhere?
Okay.
And then because I'm an experimentalist, I'll use that word. I'm not the sharpest sometimes. So I force my student during COVID to look through those 600 proteins to make sure it's there.
Okay.
So she's ready to graduate and she's like, can I please not look through anything manually ever again? But I think love computational work, think is amazing, but we have to have the human eyes sometimes to look at things. Well, for sure it's biased because the structure that we find, some of them that seem to have a bond, I don't know how much you have looked into structures, but they have an annotation in the PDB. But if you know a little bit of chemistry, you know that an amide bond cannot be formed at that pH and so on. So we excluded those and said no. So we're saying that PLTs have in general the isopeptide bond in lasso peptides, but it's formed by an enzyme that can make and break it. And those proteins for sure do not thread and unthread.
Got it. So it's a high degree of confidence. Are these all monomeric proteins or is
No.
there quaternary structure sometimes too?
Yes. So SOD is a dimer,
Yeah.
a homodimer. Chemokines can be monomers, homo, and heterodimers. So the interesting part there is we kind of want to just switch the core out and see if they can bind to another receptor, but it's a lot more complicated. They also have post-translation modifications and other things.
Thank you.
Great question.
Any questions from students? With 80 students in this room.
I have one. Hi, so I've only taken organic chemistry, so I'm pretty the protein biology, protein biochemistry. So my understanding is that like the shape of the molecule largely is due to intermolecular forces that force the rotations of certain bonds and certain positions and atoms and molecules and constituents into certain respect to one another. But when we're talking about a chain being malleable, that it can take on many different shapes, and how is that? Because that's different from what I've learned like Vesper theory.
That's also a great question. So malleable in the sense, secondary structures are secondary structures and they have the same connectivity. So if you look at this, when I say malleable, this loop can be in this position, but it can also be in that position. So it's basically, so this protein is also made of 50% of loops. So it's basically the loops, but you can see one thing that we found with NMR, but also you can read these maps. This is actually showing that little 4' helix is what we call it, because you have a 4 helix bundles, and usually the 4 helix bundles have an extra little turn or like extra little helix somewhere, which is here. And looking from NMR is actually just populated 50% of the time. So I decided not to talk too much about biology, but we believe that that has a role when it comes to binding and activating the receptor, because these transiently formed secondary structures are usually indicative of a biological influence. So when I say malleable, basically the loop that we thread has this helix, the disulfide bond, and there's a long loop, and you can see that this is basically collapsed. The structure is not from NMR, that's not the people, this is NMR.
That shape is driven by biology.
Yes, and we talked about it in the lecture this morning. They're a native state, but everything are ensembles. It's always more than one state, and that's what we mean by the malleable. The loop can be many different orientations. It's more rigid down here, right? It's more messy up here, so it can kind of fit in, but we can't, a secondary structure is a secondary structure, and most of it is determined and rigid, but they can move. That's all. Awesome.
I know you didn't talk too much about it, but with your knockout slide, why might you be surprised that the microbial diversity increased compared to a while?
I'm guessing I heard someone is doing a lot of microbiome research here. That's, we're part of this microbiome collaboration in Hawaii. I expected, because we talk about microbiome playing a role in obesity, and I expected a low diversity being more obese. So there's a lot of research showing that the microbiome composition influences obesity. So we expected if we knock out leptin is proposed to be the link between your gut and the brain. It has the full-length receptor in your brain, but it has receptors in your stomach too. It has receptors across your entire body. It's expressed mainly in adipose tissue, a little bit in the gut mucus or something like this, but there are receptors everywhere, and it's part of puberty. It's part of fertility, and it's proposed to be the link between the gut and the brain. So we thought if we knock out the signal molecule that tells the brain what's going on in the gut, we expected to decrease the diversity, but they actually increase it. But is that something that you have seen before? So to you is completely expected?
No.
Because I've heard other research, they've seen similar things. So it might be good to have or bad to have a more diverse if you have the wrong bacteria, because we haven't analyzed which ones yet. So it's great observation. It probably depends on what bacteria you have as well.
Yeah, we study honeybees, so
Oh,
any,
cool.
like disturbance of what is done naturally will increase the very disturbed. So at least in the honeybees, a knockout would probably increase the risk.
Awesome. Thank you.
Is there like a specific rule that more or less knotting and folding makes the protein like more or less grow to degradation property in it, or does it like depend on the protein itself?
I didn't talk about that because that's exactly what we think, and we don't have any experimental data yet. But for those of you who know the ER, you don't have any degradation system. So for the protein to be degraded, you have to go back to the cytosol. And it's again a little pore, kind of like a chaperone pore where you have to push it through. The question is with knotted proteins, people believe that they're knotted just to protect them from degradation. And it could be true in our case too, but then a protein that is just a circle, I would believe has as much of a problem to go through. So it could be, so we're trying to kind of understand if it's a difference between having disulfide forming loops in proteins or if the threaded element plays a role. But that is a cool project that we really want to explore.
We have a question on Zoom from a student, unless there was. Go ahead.
It's actually kind of bouncing kind of off of the last person's question. I don't know if this is something that you focused on or in this field has been focused on. Does the knotting in the folded state, does it have any effect on the rate that the protein unfolds? Is that something that's been observed or?
Yes. So in simulations, we have these events where it unfolds, but it doesn't unthread. So we think it biases the protein to the folded basin. And I think if I can quickly pull up this slide, if we can go back one. There we go. I don't know if it's visible here. If you just look at the blue and the red, the oxidized protein is slightly below. So all these events, they go all the way here, but they're always threaded and then collapse back. That's where you see that this barrier is lower.
Right.
If we exclude all those events, it overlaps with the blue. So it does actually prevent the protein from unfolding. Great question.
So the question on Zoom from Madison is sort of related. She was asking if once the end is plugged, let's say, do you have any insight into whether there's like rearrangement of side chains, for example, that kind of tend to lock it in place? The analogy she used was like kids sticking their finger in one of the straw holes on cups and then getting stuck.
Yeah, great question. So residue L51 is a disease mutation, and humans that have this mutation get severely obese, infertile, and they have all kinds of problems. So L51 is kind of the protein, the little, let's see where we have a structure here. So L51 is just in this loop. I don't know if you can see it. So this is the end terminus. Helix A comes up here. You have a really long flexible loop. You actually have a second hydrophobic core here, what we found with NMR. And then the loop comes down, and then you go through with helix B. This is 51, connecting to this amino acid here. This is a backbone link. If I mutate that and put it a serine in that they do in the disease mutation, the protein is not folded in. So it's extremely important. So they're probably doing something there. We should try and put another amino acid in. I don't think I did that, just to see, does it have to be a lysine, because it's a backbone bond. So I don't understand why a serine would not work, but it's doing something sterical.
This is a little less specific, but I saw on your timeline at the beginning that you had AlphaFold on there. So have you used that at all in your research?
I have. I think it's a great tool. When it comes to the leptin receptor, I did this in collaboration with one of my postdoc coworkers again, and they did a docking simulation. So what we found was everything is not as planned as it sounds in papers, just for those of you starting your academic career. So when I tried to solve the NMR assignments, there's a second tryptophan here, and there should be tryptophan here. To crystallize the protein, they deleted this and says it has nothing to do with activity. So I did the same here and got the NMR assignments. It took me 9 years. It was so easy to do that. It took forever. And then I got it and everything was ready to go. And then we had a collaborator do an activity assays. We just threw it in there and it's not active at all. So it's proposed to be 3 binding sites for cytokine proteins. Leptin is the cytokine. So this is called binding site 1. And everyone said that has no role. So we thought, you did a mistake, do it again, do it again, do it again. And our collaborators say, we're not going to do it again. It's not active. So if we put in anything negatively charged here, we completely abolish activity. And when we do that, it does bind to the receptor, but it does not activate it. So we did an AlphaFold prediction, and because the receptor has a transmembrane part and an intracellular part, it's really hard for AlphaFold to predict it. To kind of wrap the tail end around the receptor. But there is cryo-EM structure of a mouse protein. So we were able to figure that out. So we docked it and we simulated it, and we saw that leptin can actually form a 3-to-3 complex. That was kind of a newer thing. People say it's 2 receptors and 2 ligands. We're just saying like something else is happening because when that is negative, we bind to the receptor, but we don't activate it. Either it's one of these rearrangement that happens in the insulin receptor, or it is this adding a third ligand receptors. We did a simulation, we just have to move leptin by 6 degrees or something, and it can just go on forever. And it was a little hard to publish, but there was 2 more papers that came out with cryo structure showing that it can form a 3-3 complex. But interestingly, that tryptophan is nowhere near any of the receptors, even when we use AlphaFold to do this 3-3 docking. So I think it's a great tool, and we use it as much as we possibly can because it can help us predict these things, and then we can test it in the lab. Great question.
So if there are any remaining questions, you're welcome to come on up afterwards. Dr. Hagelin has a few minutes before her next meeting to chat with folks, so, but let's thank her for a great talk.
Thank you.
Those of you with the seminar course, there is a paper floating around.
I took-
Signing in, but please make sure it's over